T cells express a T cell receptor, which is a heterodimer, and this T cell receptor recognises small peptides bound to MHC molecules. So in the case of CD8 positive T cells, they recognise MHC class 1 presenting peptides, and CD4 T cells recognise MHC2 presenting peptides. So when a T cell recognises its target, that TCR-MHC interaction is the first step, the first sort of all-important signal. The second signal required is, of course, comes in the form of co-stimulation, which in a naive T cell is presented on a licensed antigen-presenting cell. Through the combination of these signals, that then drives T cell activation and ultimately influences cell fate in terms of proliferation and survival and various effector functions. The difference between CAR T cells and how they differ is that a CAR, or a chimeric antigen receptor, is a synthesized, modular, genetically engineered receptor which essentially uses the signaling tails of the T cell receptor, so the CD3 protein, which normally associates with the T cell receptor non-covalently. It's fused to the intracellular signaling tail with a co-stimulation domain in the one receptor, and then on the outside of the cell is a single chain derived from an antibody fragment. So a heavy and a light chain of an antibody that's fused together and fused to a transmembrane domain, and then the co-stimulation, and then the CD3 tail inside the receptor. So what this means is that now, if we can design an antibody against a protein, any protein of interest, anything you like, we can now design a killing receptor against it. So we can replace the T cell receptor with this genetically engineered receptor called a CAR. The great thing about this is that the CARs bypass the requirement for MHC class 1 restriction, which means that tumour antigens that might be presenting mutated peptides on the surface of the cell, if the patient's own immune system, if they don't have the T cell receptors required to recognise that, we can bypass the need for MHC and just design synthetic receptors which can recognise tumour cells. So why would you use a CAR engineered cell therapy over a standard TCR therapy? One is that we can design them against whatever we want. We can target very specific tumour-specific proteins. And in the case of, and it's worth keeping in mind as well that although we're talking about in the context of cancer right now, you can design a CAR T cell to target any antigen of choice, not necessarily just tumour cells. For example, I would envisage that you could use CAR T cells to also kill HIV, latently infected T cells, or autoimmune T cells. So you could eradicate any cell from the body using a CAR T or engineered CAR T approach. Quite often patients don't have the T cells that recognise their tumour. And so we see this very frequently in tumours which are not have a high mutational load, for example. So some tumours acquire a lot more mutations than other tumours. So for example, tumours like melanoma respond very well to cancer immunotherapy checkpoint inhibitors because the patients will already, in a lot of cases, perhaps have an immune response to the tumour, but it isn't sufficient, or it's being dampened down or switched off by a very immunosuppressive tumour microenvironment. So in that case, you could take a different approach. But for a lot of tumours, like brain cancer and others, they're not very mutagenic. So they're quite invisible to the immune system. So the patients don't have the T cells to even recognise the tumour in the first place. The short answer is I don't think so, but this is very cutting edge. So actually in my lab and also in the lab at Peter Mac, we've been exploring this exact question. So does a T cell with an engineered receptor function differently to a native T cell? In terms of that delivery of the lethal hit, not really. They utilise the same mechanisms of exocytosis of secretory granules to the target cell to induce apoptosis. So the mechanism of how these T cells kill is the same. From our studies, we've found that the CAR T cells that are triggered tend to degranulate a little bit faster, so they have a bit of a stronger signal coming through the T cell. But ultimately, the function is the same, and the target cells that have been delivered a lethal hit take the same amount of time to die. So now, whether or not the function of CAR T cells and native T cells differ in other characteristics in terms of their persistence, their ability to persist, and form memory cells in the host. Whether or not they become exhausted or anergic or get sort of shut down if the CAR T cells are being overstimulated. And then what is the consequence then for release of cytokines and other effector functions is, I think, really yet to be fully explored, and it's something that we're very interested in studying in my lab.